Well, today we start a first unit. It's organized in, I believe, 6 units, the whole class. First unit will be about endocrine system. So, endocrine system, unlike many other body systems, which are comprised of connected organs. Look at in the corner of the class, where there are those glass boxes with cardiovascular. You see all the blood vessels and the heart are connected, right? In endocrine system, you don't have a lot of connections between different organs. They aren't physically together. They kind of spread throughout the body, located in different parts of the body, but they all perform the same general function. They regulate, regulate body function. And what I want to highlight, this regulation is completely beyond your conscious reach. You cannot, by any means, control the hormones. This is truly unconscious, involuntary responses. Now you may say, wait, but we have nervous system, and nervous system also controls this unconscious, involuntary responses. That is true. Endocrine system, nervous system both participate in regulating your visceral functions. Remember, visceral organs. But they do it in a different manner. This table compares how these two systems are different. Take mediators. In the nervous system, we call these mediators, these chemicals that are released from neurons. Can anyone remind me? How do we call them? Neurotransmitters, right. So these neurotransmitters are released from neurons, right? And they're released locally. So if you have a neuron, coming into the organ, that part, that axonal branch, will be the only place where neurotransmitter is released. So they local, and they respond always. Neurotransmitter is released because of the electrical impulse, right? Now, if you think about endocrine system, release of hormones can be stimulated by changes in the internal environment, like elevated or decreased glucose. Can be stimulated by nerve signal, or it can be stimulated by other hormones. And that release of hormones are released in the circulation, in the blood. And then blood delivers hormones to virtually all the cells in the body. So nervous responses, nervous signals are local. Endocrine signals are systemic. And that defines, the site of action. For nervous system, it's always a synapse. Action will happen at the synapse. Signal will be transmitted at the synapse. Now, in endocrine system, insulin that is released from a pancreas will get in the blood, will reach the brain, and will act on the brain cells to increase the glucose uptake by those cells. Does that make sense? For nervous system, what are really the target cells? Nervous system, what are two effectors of the nervous system? Muscles and it's like a rhyme. Muscles and glands, right? So nervous system can stimulate muscle cells or glands or other neurons, yeah. Endocrine system, insulin can target muscle cells, neurons, hepatocytes, adipose cells, cells that have no real resemblance but they all be targeted by insulin. So they're much more variable in terms of effector cells. And of course, nervous responses are really, really fast. They're also short. Does that make sense here? So, nerve signal will deliver the response, so time for action is really short, but effects are short-lived as well. In case of endocrine system, responses are pretty long. You will see that when we say we're going to talk about glucose homeostasis, and you will see the effects of insulin, effects of glucose on the insulin release. It takes time. Okay, and it takes time for the glucose to be taken out of the circulation. It takes minutes, sometimes hours, sometimes days, but the effects are long-lasting as well. So you see, as we saw before, it's kind of a repetitive theme here. The effects, if effects are fast, they're usually short. If effects take a long time to develop, they're usually long-lived. And one of the major differences, nervous system is more local, while the endocrine is more systemic. Now, if you want probably one of the most important concepts that will appear over and over as we talk about endocrine system is the idea that hormones can act on the cells by interacting with the receptor. Hormone doesn't just directs the signal. Hormone has to bind to a cellular molecule that we call a receptor in order to initiate that signaling. So far, any questions? We all good and clear? Okay, few words about endocrine anatomy. Endocrine anatomy, studying it is like taking candy from a baby. It's easy. There are two types of endocrine organs: primary and secondary. So primary endocrine organs include, and I go from top to bottom, hypothalamus, pituitary gland, then pineal gland, which I can't, like, it's really here, really tiny, thyroid gland, thymus, adrenal glands, pancreas, and gonads. This is it. These are all primary endocrine organs. When we use the adjective primary, we mean that the sole function of these organs is to produce hormones. Am I clear? They don't do anything else. They just make hormones. Unlike primary endocrine organs, secondary have some other major function. For instance, heart pumps the blood, but also produces a hormone known as atrial natriuretic peptide. Liver is responsible for metabolizing different chemicals, but produces such hormones as thrombopoietin and angiotensinogen. Adipose cells store energy, store fat, also producing hormones such as leptin and adiponectin, which we're going to talk about. We're going to have a little conversation about secondary endocrine organs. Everybody's good. You understand the difference between primary and secondary. We're all good. We're all set. Okay. Now, all hormones can be divided into two chemically distinct groups. water-soluble, which are highlighted in blue, and lipid-soluble, which are highlighted here in red. This separation is really important because the solubility, as we will see, defines the location of the receptor for the hormone, and therefore defines mechanism of action. Now let's take a glance at three categories of water-soluble hormones. Those are amine hormones that are usually derived from amino acids. Okay? Amino acids that are building blocks of the peptides or proteins. Therefore, peptides can be water-soluble hormones, such as oxytocin. And finally, hormones that are proteins. All these three kinds are water-soluble molecules. Question? We're all good? Now, you do have to know which three types of water-soluble hormones there are. Amino molecules or amine hormones, peptides, and proteins. Am I clear so far? Among lipid-soluble hormones, there are three major classes. First class is steroid hormones, that's structurally based on the molecule of cholesterol. And this family includes such molecules as estrogen, testosterone, cortisol. We're going to talk about these hormones. Thyroid hormones, shown here. Little disclaimer, sort of a, not really diversion, but you do not need to know a chemical structure of a single molecule in this course. I will not ask you about any structural formulas. Okay? So, thyroid hormones are also lipid-soluble. And finally, eicosanoids. I don't talk much about eicosanoids for one major reason. I am really scared to talk about those because there are dozens and dozens of molecules with various effects, but these effects mostly associated with inflammation, but they can entail regulation of the metabolic responses. I really don't want it can be a separate course, derivatives of arachidonic acid, which I don't want to do. So we're going to focus in terms of lipid-soluble hormones. You do have to know these three families, but in our further conversations, we're going to be talking about thyroid hormones and steroid hormones. You're all good? Moving on. Let's talk about the mechanisms. So, what is the reason I obviously pay so much attention to the hormonal solubility? For this, we need to quickly recall the structure of the cell membrane, which is phospholipid bilayer. Can anybody tell me which molecules can cross phospholipid bilayer easily and quickly, and which molecules cannot cross phospholipid bilayer unless they are heavily assisted? I'm talking about hydrophilic versus hydrophobic. Which ones can cross and which ones cannot? The opposite. Think about this. The phospholipid bilayer is hydrophobic on the inside. So hydrophilic molecule cannot get through. water-soluble hormones. They're massive. Look, it's a protein. It's a freaking protein. It cannot get into the cell easily. So they will stay outside. Hydrophobic molecules, such as lipid-soluble hormones, can fairly easily cross the membrane and get into the cell. Now that entails a fundamental difference in the location of a receptor. Remember, we talked about hormones interacting with the receptor just 5 minutes ago. So think about this. Let's talk about water-soluble hormones first. Water-soluble hormones, here, they stay outside of the cell. Is that clear? We're good? They stay outside. Therefore, the receptor has to stick outside of the cell. So receptor for the water-soluble hormone will be found in the cellular membrane, on the cell surface. Do I make sense? It's going to be a transmembrane protein sticking out. That's kind of a receptor, okay? So you see this part, the part where hormone binds, it is sticking out. So when hormone, look, when hormone binds to the receptor, when hormone-receptor interaction occurs, receptor, this or this, initiates some kind of intracellular signaling. You encountered that before. Do you remember second messenger system in the nerve system signaling? Second messenger in, in the retina, phosphodiesterase mediated, second messenger system in the taste sensation, second messenger system in the olfaction. All those situations when you have a hormone or, I don't know, molecule that binds to the receptor on the surface. It's like you knocking on, like a postman, okay? Like a postman knocks on your door. You don't let them in. You open the door. They give you a package and they leave. In this case, you receiving the package is the receptor. And the postman with the package is the hormone. Does that make sense? We're clear. Passing of the package.
Of the long, what was it, the meter stick that we had last semester, whenever you'd say, I have this and I'm giving it to, I think it was like Simone, I think in our class. You said she takes it and then she gives it to somebody else. Or
sort of, yeah. It's like second messenger system. So, say, somebody, I get the package, but it's not for me, it's for my wife. So I'm a receptor. I merely receive that interaction with the postman. Then I turn around and give it to my wife. Okay? So I initiate intracellular signaling. Does that make sense? The same happens here. The whole function of a receptor is to start some kind of an intracellular signaling. There are 3 mechanisms that you must know. Only 3. As I mentioned, everything that you need to know I will tell you. So first mechanism is right here. You're familiar with that? It's called adenylate cyclase mediated mechanism. Here you have a receptor. Okay, and this one here is a G protein. When hormone binds to the receptor over here, this binding immediately activates G protein, which then activates the molecule shown here. It's called adenylyl or adenylate cyclase. Adenylate cyclase facilitates a very simple reaction. It converts a molecule of ATP into a molecule of cyclic AMP, or CAMP. And that CAMP serves as the second messenger for many events downstream. You need to know this pathway till CAMP. CAMP can initiate this protein kinase, this protein kinase. Protein kinases, does anyone remember what they do? MLCK, myosin light chain kinase. What do they add? Which residue do they add to other proteins? Kinases. Start with P, PH. Phosphate, yes. Kinases, they add phosphate residue to other proteins, activating them. It's basically like a game of tagging, okay? G protein here activates adenylyl cyclase. Adenylyl cyclase produces CAMP, and then CAMP starts to activate proteins down the road. This, that, this, that. Those proteins will activate other proteins and other proteins. That makes sense here. Now, one little thing about, like, let's imagine the signal. So one single receptor, which interacts with one single hormone. Are you with me? That activates one single G protein, which then activates a molecule of adenylate cyclase. Now, adenylate cyclase will start producing cyclic AMP. Is it going to produce just one, or it's going to just keep churning them out? It's an enzyme, so they're going to keep coming. So now we're talking 10, 50, 100, 1,000 cyclic AMP molecules. Each cyclic AMP molecule, CAMP molecule, can activate 10, 50, 100 different proteins. Each of these proteins can activate many more proteins down the road. Does that make sense? So it grows like an avalanche. You got it? This signaling is really fast. Okay? Because it does grow like an avalanche. It's really, really fast. That makes sense so far? So again, as I told you, I am at liberty to ask you about any step up until cyclic AMP is synthesized. What happens next stays in VEX. Okay? I'm not really interested because there's so many possibilities. Forget it. Am I clear? Any questions?
Where is that?
CAMP?
Yeah, where is that?
It is synthesized by the enzyme called adenylyl cyclase. Can you see that?
Yeah, so we don't need to know all 6 steps, just all the way up to...
Oh, no. I just need to know, okay, we got CAMP. CAMP can activate a lot of proteins downstream, period.
Okay.
Period. But I can ask you, which enzyme synthesizes cyclic AMP?
Okay.
Or, what is the function of adenylate cyclase? Or, G protein, in this case, well, it's kind of obvious, but, you know, activates. If I ask you, G protein, what does it do? Does it activate adenylate cyclase or produces CAMP? You tell me, no, it activates adenylate cyclase. Does that make sense? Up until CAMP, I can ask whatever. Any step I will require. Beyond that, nothing. We're good? Phospholipase C. Now, this is very, very similar. In this case, you're already familiar. Look at this. You have a hormone. Can you see that? It binds to the receptor. That binding, again, activates G protein. Activated G protein activates another enzyme that is called phospholipase C. And phospholipase C produces the molecule called IP3, So this is going to be your second messenger. That makes sense so far. IP3 then binds to the receptors on endoplasmic reticulum and releases calcium. Now let's do it this way. I'm going to ask you a question, or I'm going to make a statement and you're going to finish it. Hormone binds to what? Receptor. Speak up. Receptor activates what? G-protein. G-protein activates what? Phospholipase C. Phospholipase C produces what? IP3, and IP3 stimulates the release of calcium. Calcium can have many effects on the cellular processes. It can open, close channels, activate different signaling cascades in the cell. We don't talk about it until calcium. Clear? Any questions? Go ahead.
How would you know if when the receptor binds if it's going to be phospholipase C or if it's going to bind to the
You're talking about whether I'm going to ask for a hormone. I will not. What I can do, let me explain. I give you a hormone, say, I'm telling you that glucagon is a protein, water-soluble, right? Which of the following signaling events can or cannot happen? So if I give you a list and one of these events, hormone will go directly in the nucleus, you tell me it's water-soluble. It's not going to happen. Or I tell you glucagon, right? Water-soluble hormone. Can it bind to the receptor in the cell membrane? Yes. Can it initiate G-protein mediated response? Yes. I will not ask you what is the signaling cascade for specifically glucagon or epinephrine or norepinephrine, I don't know, insulin. Does that make sense? You really need to understand where the receptor is. Am I clear? In general, well, you don't have anything to refer to yet. But I always say, ask me, always ask me. But if something in the study guide, you should know that. Okay. Now, the third mechanism for the water-soluble hormones, and those are three mechanisms that you need to know. I'm not going to ask you about any examples or such. Is receptor bound tyrosine kinase. So this is the receptor. Can you see the receptor right here? This is a hormone. When hormone binds with the receptor, it activates the enzyme right here that is called tyrosine kinase. Tyrosine kinase, as all kinases do, adds a phosphate residue to a protein. And the phosphate residue is added on the tyrosine. And then think about phosphorylation as flipping a light switch. You understand what I'm trying to say? You had an inactive protein. You phosphorylated it, you flipped the switch, it's an active protein now. Does that make sense to you? We good? You see, there's no really a chain. The kinase is basically attached to the receptor. Yes?
What's the point of the phosphate residue?
Great question. So phosphate residue is negatively charged. Does that make sense? So far, give me a sign that you follow. It's negatively charged. When you put a group with a negative charge, 1, but it's a small group, you're going to put a small group with a negative charge onto the protein, because of that negative charge, protein will change its shape. When structure changes, what changes as well? Function. So it's a way to change the protein structure to make it active. Good? Yes.
You said that hormone signaling in a whole is super fast.
It's not.
Well,
Go ahead. Keep going. Ask your question. I don't want to
I just want
interrupt.
to make sure that whenever, I think it was back whenever we were talking about the very first one, we said the signaling is really fast. I just wrote that down. I just wanted to make sure it wasn't just that one in particular, but all hormone signaling.
Thanks. Great question. To clarify on the fast signaling. Water-soluble hormones, whether it's whether they work through adenylate cyclase or phospholipase C or tyrosine kinase, they all initiate an intracellular signaling that develops like an avalanche, like a cascade. Does that make sense? So, in all these cases, signaling by hormonal standards will be pretty fast. Does that make sense? Not as fast as the nervous, but pretty fast. Am I clear? Good? Because what we compare it to? We compare it to the lipid-soluble hormone signaling. Now, lipid-soluble hormone can cross a plasma membrane. We got it? When hormone crosses the plasma membrane, here's the hormone, it enters the cytoplasm. And then there are two options. Different hormones use different pathways. Hormone can either bind to the receptor in a cytoplasm, or in some cases it can bind to the receptor in the nucleus, depending on how far the hormone goes. Does that make sense? The most important thing is that receptor here is inside of the cell, whether it's a cytoplasm or nucleus. Are you with me so far? So the complex between the hormone and the receptor will inevitably be in the nucleus. Whether directly binding will occur directly in the nucleus, or this complex here will then move into the nucleus and will bind the DNA. I specifically highlighted this. It's the hormone receptor complex, not the hormone itself, that binds the DNA. Am I clear about it? Yes?
So what you're saying is, for like steps 1 through 4, you're saying it could go from either 1 to 3, or it could go 1, 2, and then 3? Is that what you're trying to say?
No, that's not a perfect illustration. What I'm trying to say is that in this image, in this illustration, hormone and receptor, on this illustration, the receptor is where? In a cytoplasm, right? Are you with me? But in some cases, the receptor can already be in the nucleus. So hormone has to go in the nucleus and bind receptor there. Does it make sense?
So in this picture, the receptor for the 2, it's in the cytoplasm.
Yes.
And then when it's talking about 3, it's already in the nucleus.
On this specific illustration, receptor in the cytoplasm. Are you with me? Hormone binds the receptor in the cytoplasm. And then this whole complex moves on into the nucleus. Bear with me. In some cases, that is not shown here. The receptor is in the nucleus. So hormone goes directly there and binds the receptor in the nucleus. But either it's this picture or the situation that I just described, hormone receptor complex will inevitably end up in the nucleus. We got it? This complex binds to DNA, and that complex binds to DNA, not a hormone itself, and that binding starts the transcription, synthesis of RNA, and then that synthesized RNA is translated into a protein, or should I say multiple different proteins. Does that make sense here? We're good. So let me put it this way. There's a hormone. I'm kind of running ahead of the train, but I just want to illustrate. There's a hormone called aldosterone. It's the fat-soluble hormone. So when it forms the complex with a receptor in the nucleus, this complex binds to the DNA, and it initiates the synthesis of a protein that is essentially a sodium channel. When you get more sodium channels in the kidneys, more sodium will move from the filtrate, from the urine, back in the blood. Does that make sense? And that's what aldosterone does. It increases the reabsorption of sodium, the retention of sodium, by stimulating the production of a protein, of a sodium channel. Does that make sense? Now this response, as you can see, it's not like cascade, like an avalanche. It is very targeted. Only certain proteins will be made. So this response is usually slower. That makes sense? Response to fat-soluble, lipid-soluble hormones is usually slow. Make sense so far? Any questions? Now, this is just to illustrate what can stimulate the release of hormones. I want to tell you, you know a lot about endocrine system without realizing it, just from your day-to-day experience of conversations. So there are three stimuli, the hormone production. First is humoral. The word humoral means fluid, liquid. So it's a stimulus that has something about your blood or extracellular fluid. In this example, it's a release of parathyroid hormone. Probably remember, this hormone regulates calcium balance in the blood. When calcium in the blood goes low, parathyroid hormone is released by parathyroid glands and moves calcium from the bones into the blood again. Does that make sense? Now here's another example. I don't want to torture you trying to, take it out of you, the example. Insulin. What stimulates insulin release? High blood glucose, yeah. So you get something changes in the blood, insulin is released. Does that make sense? So this is your humoral response. Humoral stimulus. Am I clear? Neural stimulus. You all remember epinephrine, norepinephrine. You get nervous signal from sympathetic activation, release of epinephrine, norepinephrine. That make sense? Have you heard the expression, maybe in the popular media, the stress hormone? Have you? Do you know which hormone that is? Yes. Cortisol, exactly. Now the stress, is it a nervous stimulus? Yeah. Look, for instance, like for me, talking to you here, I'm used to it, but I know that many people are really, really stressed from public speaking. Okay? So my levels of cortisol now are probably normal, if somebody who's not like really shy will take over my place, that would be terrible stress for it. So that's nervous stimulus. That makes sense? And finally, one hormone can stimulate other hormones. For example, many hormones that are released from hypothalamus stimulate the release of other hormones from the pituitary gland. You got it? So this 3 responses now. I can give you a description for, I don't know, aldosterone, insulin, whatever. I'll give you a description what stimulates the release of a certain hormone, and you will have to tell me, is it a humoral stimulation or neural stimulation or hormonal stimulation? Am I clear? Any questions? So hormones, just a reminder, first, autocrine, paracrine, endocrine signaling. So what is autocrine? Autocrine signaling is the signaling on the same cell. Look at this. You see, cell releases something, and then this something acts on the same cell. That makes sense? Just by definition. Paracrine signaling. Hormones act on the adjacent cells. And finally, truly endocrine signaling. Hormones get in the blood and move all the way someplace else. As I mentioned, insulin that is produced by pancreas can act on the neurons in the brain. Okay. Antidiuretic hormone that is released from pituitary gland acts on the tubular cells in the kidneys. That's endocrine. That's PAR. Any questions on the modes of actions? Basically, just know the definitions. The regulation of hormonal action. It looks like it's not really an important concept, but it is. Hormones are regulated by the principle of negative feedback. So I want to first do it with you without any real examples. Okay? So something changes in the body. Something changes. In response to that change, the hormone is released. If it's a negative feedback, what should this hormone do? Should bring it back to balance, should basically cancel out this change. Does that make sense? When the change is gone, what happens to the hormone production? Stops. Yeah. That's the whole story. So this picture here shows you that exemplified by cortisol. Say levels of glucocorticoid, cortisol, in the blood go down. What kind of stimulus that is, if it's in the blood? Huh? Humoral. Excellent. Humoral stimulus. It goes down. This humoral stimulus is sensed by hypothalamus. Hypothalamus. Which releases a hormone called CRH. Corticotropin releasing hormone. Now, corticotropin releasing hormone acts on the pituitary gland, now leading to the release of another hormone called ACTH, adrenocorticotropic hormone. Now that's signaling when one hormone leads to the release of another. This is an example of hormonal stimulus. That make sense? ACTH stimulates adrenal glands to release cortisol and levels of glucocorticoids in the blood go up. So they go up, the whole signaling stops. Does that make sense? This whole negative feedback thing? Are we clear? You're going to see it over and over. You get used to it after like thyroid hormone, it will go like this. There are two examples that I know of positive feedback in hormonal regulation. One, you well aware of, oxytocin during birth. You know, more oxytocin, more uterine contractions, more pressure on the uterine cervix, more oxytocin, the more uterine contraction goes this way until baby is born. Okay, and the second example of positive feedback, it's a short-term positive feedback in the female reproductive system regulation. Right before the ovulation, estrogen and luteinizing hormone establish positive feedback loop that eventually leads to the ovulation. We're going to talk about it in a separate occasion. Any questions? We're going to take a break. We'll come back. We're going to chat about some other hormonal aspects. I'll probably give you a first lab today.